Welcome back. Nice to see you all, and we go on to the afternoon session, and it's a great pleasure to begin by introducing Mary-Ellen Lynall, who, as you can see, is academic ACF at the University of Cambridge, also is academic foundation fellow in her foundation years, and as you can see from her CV, she's almost as sickening as Carl's, and indeed in a few years' time it will be as sickening as Carl's, deciding which paper shall I publish in Nature this week. But what you don't know, but I know, is she's also very typical of psychiatrists, because many psychiatrists are people with what Dennis Healy used to call a hinterland. There's more to them than meets the eye. And Mary-Ellen is an extremely distinguished musician. She's a choral scholar at Clare. She's a very good, obviously a very good violinist, played in the National Youth Orchestra. And I mentioned that because my mother was one of the founders of the National Youth Orchestra as a violinist in 1945. It's a little known but absolutely true fact. She's also a singer, and I'm pretty sure, Carl, you haven't got this reputation. She's sung Mabel in The Pirates of Penzance. I can't quite see you doing that, but you never know. Kate in Kiss Me Kate, I mean, I know you're a talented man, Carl, but I really can't see you have the upper register for that. You played the lead in Hindemitz's Murder, the Death of Women. Oh, we've got a mic now. That makes a change, doesn't it? Well, anyway, known as The Woman and The Lady in something by Handel, and so on and so forth. And Velma in Bugsy Malone. I rather like that. Good. So, remarkably talented people we get in psychiatry. Mary-Ellen, the floor is yours.
And I don't think we should doubt Carl. If he wants to sing Mabel, I'm sure we can work on that together. So thank you all thank you for inviting me. It's an honor to be up here with this incredible lineup. I'm going to talk to you today about the immune system and stress and depression. So I'll tell you a little bit first about why psychiatrists should be interested in the immune system at all, give you a bit of a recap of the immune system basics without post-lunch sending you to sleep too much on undergraduate immunology, and then talk about the mouse models of stress I've been using to unpick how the immune system and stress are related, and then talk about a little bit about the human clinical biomarker studies we're working on at the moment as well. So you've probably heard of immunopsychiatry. There's a huge impetus behind it at the moment, and you can see from the publication record that there's an increasing interest in this area. And that's in large part because of the potential for new therapeutics. So why do we think it might work to seek therapies targeting the immune system? Well, we've had about 20 years of huge investment in novel pharmaceuticals targeting the classic monoaminergic and other neuronal targets, and that hasn't really worked out. We haven't had any novel classes of antidepressant rationally based on that research. But the difference with the immune system as a target is that we've got easily accessible biomarkers. So with a simple blood sample, you can get an incredible readout on what's going on with the soluble and cellular components of the immune system. So we can, if you're looking at a serotonin receptor in an individual patient, there's no easy way to see, is there a problem with serotonin receptor expression? And with our treatment, have we changed that at all? Whereas with the immune system, simple blood test, you've got your biomarker, you can see the pathology, you can select the patients which have that pathology. Stratification is going to be really important here. And then you can see if there's a response in the biology in the early stages of your treatment, which will considerably ease the progression of compounds through trials. And what would success look like? Well, no, we're probably not going to be replacing standard antidepressants as a first-line treatment in psychiatry. It's more likely going to be that treatment-resistant group who have maybe raised inflammatory markers who will be able to select for immunotherapy. And hopefully we can repurpose some of those therapies used in other areas of medicine by the rheumatologists, by the renal physicians, and use those to help some of our patients with depression. So let me just take you a little bit through the evidence that tells us that there is a link between the inflammation and depression. People started out by doing really very simple case control designs, asking, is there a change in the soluble components of the immune system in depression? And as you can see from the dates, this is an area that's been of marginal interest in psychiatry for over 20 years now. So CRP is one of the soluble components. It sticks to pathogens, it targets them for destruction. interleukin-6 and TNF-alpha, they're pro-inflammatory cytokines, and they're released by all different types of immune cells, B cells, monocytes, T cells, and they propagate the immune response. And what we can see is that these data over many years have converged on the solid but quite limited finding that, yes, these things are slightly higher in depression than in people who aren't depressed. And that's something we see in our local data as well. So this is C-reactive protein in the Wellcome Trust-funded BioDepth study. And you can see that it is raised in depression, and particularly so in that treatment-resistant group. So that's in the blood. What about in the brain? Well, in this study, the authors used a PET ligand to look for microglial activation in the brain. And what they showed was that, indeed, in people who are depressed, there is greater microglial activation, and that's particularly true in those areas of the brain involved in mood, the anterior cingulate cortex, for example. So there's good evidence of an association here, but is this inflammation a cause or a consequence of depression? So one study which speaks to this is the RCPsych cohort. So in the RCPsych, which is a very large cohort study looking at a number of different outcomes, including depression, they looked at IL-6 levels in the serum of kids aged 9. They divided those kids into 3 groups based on whether they had high, medium, or low levels of IL-6 expression. And it turns out that those who had the highest level of IL-6 when they were 9 had a much greater chance of going on to be depressed at age 18. But where is this inflammation coming from? There's no underlying infection here or autoimmune disease. These are otherwise healthy people. And one of the things that both increases inflammation and affects mood and risk for depression is stress. Now, so this is a study looking at teachers, and the authors measured the monocyte production in the blood of IL-6 from these teachers. And those teachers who were stressed and burnt out had higher levels of IL-6 production than those resilient teachers. And then they made them do a stressful test. They had to do some public speaking. And after that, they took their monocytes, like my monocytes in 20 minutes, they were producing more IL-6. And that was particularly true of those burnt out teachers. Now, we've talked a lot about the soluble components of the immune system, these pro-inflammatory molecules. But what about the cells? Well, we know much less about this because it's only really been in the last 10 years or so that we've developed the tools for looking at cells with the clarity that we can now. But what we do know is that in people who are depressed, especially severely depressed, they've got almost double the number of B cells in their blood compared to healthy people. And then this study on the bottom here, this is stressed doctors on and off duty on intensive care shifts. And these doctors, being stressed, cause a rise in neutrophils, monocytes, lymphocytes. So all of this that I've told you, here you can see a summary of what we know is going on in humans in the immune system in the periphery in depression. This raises the possibility that protecting the brain from all this nasty inflammation could actually improve symptoms of anxiety or depression. And this could be true both for, inflammatory diseases associated with depression, such as rheumatoid arthritis, ulcerative colitis, and it could be true for primary progressive disorders that we see. There's only been one trial so far that's actually looked at whether targeted anti-inflammatory treatment in depression could improve outcomes. So that's this study. It's a trial of an anti-TNF molecule called infliximab. What they did was they took people with treatment-resistant depression and they gave them infliximab versus placebo. Now, overall, there was no effect, but the interest comes when they divided up the participants into by their CRP level. So what you can see is that those participants with low CRP, actually the infliximab made them worse. It wasn't a good treatment. They had no evidence of inflammation in their blood, so maybe it's not surprising. But in those participants with a high CRP, they got better with the anti-TNF treatment. So that's kind of a summary of the background of when I came into it, what was out there, the most important findings. And because this is a conference about training, and I know we have a few medical students in the house, so I thought I'd tell you a bit about how I got into this area and why I became interested in it. So it started when I was 4. Somebody gave me this book. I don't know if any of you saw this, Cell Wars. I loved it. Attackers, defenders. It was like this guerrilla warfare going on. Anyway, long story short, I went to Cambridge to do neuroscience, and I was kind of thinking about other careers, maybe Simon alluded to, until I did my undergraduate research project with Ed, and I was just really gripped. I mean, Ed was a very inspirational supervisor, but also I was very interested in this idea of that we could develop biologically based treatments for psychiatric disorders. And we were working on schizophrenia and functional connectivity, so I was reading a bit about schizophrenia, and it came up that the main risk factor genetically was the MHC locus, a group of genes in the immune system. So I was reading a bit more about inflammation in psychiatry in general and saw this evidence which was really intriguing. And looking at what was out there, it seemed to me that there were these interesting associations, but what was missing was mechanism. So I really wanted to work out how these changes in the periphery were linked to mood and anxiety. I thought I'd be in a better position to do this as a doctor, so I did graduate medicine at Oxford, and then I came back to the east of England to do some rotations in neurology and neurosurgery and to work with Ed again. Then I was looking for an immunologist, so it was hard to find a friendly immunologist who could see the potential of immunopsychiatry. The immunologists tend to think that they're a lower-hanging fruit, but I was fortunate to find Mena Clatworthy, a renal physician, who is now my co-supervisor. So after that, I've now got an academic clinical fellowship in psychiatry, and I've developed a program of human and animal experiments trying to address these questions about mechanism. I have a small grant from Anne Charitable Trust to support my work, and as part of that, I've been able to spend a few months at the National Institutes of Health in Washington working on some of these studies. So, the immune system bit. Let's recap some of the main players. So as you probably know, there's the two arms to the immune system, the innate and the adaptive. Now, the innate immune system, that's your first response to infection. It's got soluble components, and it's got these cells. It's non-specific. And in your blood, the main phagocytes that engulf cells and debris are the neutrophils. And in your tissues, the main phagocytes are the macrophages. In the blood, there's also the monocytes, which get recruited to tissues during inflammation, where they can differentiate into macrophages. And in the brain, the macrophages are microglia. And on the adaptive side, so that's the specific response. So T cells and B cells recognize antigens, which are molecular shapes particular to a certain pathogen. They divide and form an army of cells specific for that antigen and cells expressing it, and then attack it. Cytotoxic T cells can kill cells directly, the helper cells can influence the response by secreting cytokines, and the B cells are best known as antibody factories, really. I've got to tell you a little bit more about B cells for reasons you'll see later. So B cells are inflammatory not only in producing antibodies, but also they have other roles, like they present antigen to T cells, and they secrete some of these cytokines I've been telling you about. But there's basically good B cells and bad B cells, two sides of the same coin. And so in the immune system, you've got to have mechanisms which cause inflammation so you can fight infection, but you've also got to turn that inflammation off after you no longer need it, it's no longer necessary. And these regulatory B cells do some of that turning off, and we can detect them by the fact that many of them express CD9, this surface marker. So they have their effects by secreting IL-10 and the influence of IL-10 on other cells in the immune system and by contacting cells directly. And these turn out to be really important. So they're a hot topic in other areas of medicine, particularly because they're important in preventing transplant rejection. And in models of multiple sclerosis as well, we're finding that they're important in preventing immune activity in the brain. So coming back to our actual question, what's going on? How is this peripheral activity in the immune system affecting the brain? Now, this was a real puzzle for a long time because we thought that we had this impermeable barrier separating the peripheral immune system from the brain, the blood-brain barrier. But over the last few years, this Berlin Wall has come down, and it's turned out that it's just not true. That although there is some separation, there are multiple ways by which the brain can influence the periphery and vice versa. And one of those ways is via the meninges. So for a long time, we thought the brain didn't have an immune system, but it does. It just happens to be around it rather than within it. So all the major immune cell subtypes can be found in the lining of the brain. And we've got some proof of concept that this is important for behavior. This is the Morris-Watermaze task. in which a rodent has to learn to find a submerged platform in murky water. And you can see that, well, during this task, T cells are recruited to the meninges where they secrete IL-4. And if you take a mouse without IL-4, a genetic knockout, it's unable to do this task. But if you give back those normal T cells to that knockout, it can do the task again, suggesting an importance of these meningeal IL-4 secreting T cells. I think this is an important study because this is a role for the immune system that's nothing to do with defense against infection or injury, ostensibly. It's about the immune system in normal behavior. So this got me to wondering, could it be that the meninges are in fact the link between these changes we see in the peripheral immune system and anxiety and depression? So obviously I'd love to look at this in humans, but various people have told me that it's not going to fly with ethics committee and wouldn't get too many volunteers for stripping their meninges off. So in the first instance, I looked at this in an animal model. So how did we get to the meninges? Well, this is a view down my microscope, and you can see that this is the inside of the mouse skull, and you can peel the dura off the inside of the skull, and then you take the brain and you pick the pia and the arachnoid off the brain. So it's all pretty laborious, and that's the reason we haven't really seen the immune cells before, is because if you just take the brain out of the skull, you're not going to get this material. And you can look at the meninges using confocal microscopy. So here you have, you might remember from your undergraduate neuroanatomy, the venous sinuses, and then also the arteries. And in close-up, here you can see, again, the blood vessels, nuclei in blue, and in light blue here are the B cells. So we can confirm that there are B cells passing through the blood vessels, but there are also B cells behind that blood-brain barrier in the meninges themselves. So what's the model of stress we used? So this chronic psychological stress model, in which you have a large dominant CD1 mouse, let's call it the senior professor of psychiatry in your department, and you have a smaller wild-type, lesser spotted junior doctor mouse, and you house them together, and then for 5 minutes every day, we raise the partition and they interact, and rapidly the lesser one learns that it's inferior and adopts a submissive posture like this. So you do that every day for 10 days, and at the end, you get an uncharacteristically sad Mickey Mouse. So how do we test this? We use a couple of tests of anxiety, the light-dark box and the open field arena. So a mouse will have a tendency to hide in places where it's safe, in the dark area or around the edges of the open field arena, but like humans, it's curious, and it also has a tendency to explore into that light area or into the center of the open field arena. And we use that as a marker of anxiety, how much time it spends in the safe versus the unsafe areas. Now, I showed you the confocal pictures of the meninges, but with that technique, you only get to look at a couple of cell types at once. And what we really wanted to do was take a more unbiased approach and look at all the different cell types we could and ask how they were changed by stress. So for this, we used a technique called flow cytometry. So I took the meninges, mashed it up into a single cell suspension, labeled it with 15 different colors of dyes, each a different color, and labeling a specific different surface marker. And then you run those cells through a flow cytometer, which gives you a readout for each different cell of what surface markers are expressed. And then on the right here, these are the plots that you get out of the machine, and they show you each dot is a cell, and by looking at the levels of expression of these different markers and the combinations of those markers, we can detect what kind of immune cell it is, and then count how many of each immune cell type there are, whether they're activated or not. So what was there? This wasn't known before. What we found that it was predominantly B cells, some microglia, some neutrophils. Back to our question, what was the effect of stress on this meningeal immune cell compartment? I'm going to show you a couple of these plots, so I'll just explain. On the x-axis here, we have the size of the change, whether these cells were increased or decreased in stress. On the y-axis, we've got significance, so the cell types which were significantly changed are in red there and labeled. So what you see is that the meningeal immune cells are reacting to stress. There's this increase in monocytes and a marked decrease in the B cells. This tells us that the immune cell compartment is reactive to stress. Now, that's behind the blood-brain barrier. Oh, and also note, what are these B cells like? Well, in stress, they're more activated than they are in the healthy mice. And there seems to be quite a local effect. It's not as if that there are changes in the spleen that are simply spilling over into the meninges, because if we look in the spleen, we don't see this activation. So it seems like there's a local process going on here. Turning to the spleen, we wanted to find a way to look outside the brain in the peripheral compartment like we've seen in patients with depression. So what is the effect of stress there? Well, you can see that, as was known previously, there's an increase in neutrophils, monocytes, macrophages. But because we looked at all these different cell types, we also found that there was an increase in B cells in the periphery. So particularly for plasma cells, and then these CD9 positive B cells, which, if you remember, were that regulatory B cell type which dampened down inflammation. So what it seems we're looking at here is that when the body's stressed, it turns on these pro-inflammatory mechanisms, but it also turns on these, if you like, homeostatic regulatory mechanisms that dampen down the immune response that it's just turned on. So you've seen that under stress, both the meningeal and the peripheral immune cell compartments are changing, but not in the same way. But the important thing here is causality, really, isn't it? How do we start to tease apart what's causing what here? Now, this is a question which perplexes everyone in the field a bit, because particularly in humans it's difficult to do this, but the animal model allows us to start picking this apart a bit. So it could be, and we know that this is true for monocytes and neutrophils, that the brain perceives a social stressor. It sends a message through the sympathetic nervous system and tells the periphery to increase the numbers of that immune cell. And it seems that might be what's going on in terms of why the B cells increase in number. We know that B cells have receptors for noradrenaline, which the sympathetic nervous system releases. And I had a look at what noradrenaline does to B cells in a dish. These are human B cells. You can see, well, it had no effect on T cells and caused a marked proliferation of the B cells. So that's in that direction. But what we're much more interested in is whether these B cells are actually making any contribution to that anxious phenotype. Because if they're not, if it's true that it's just brain is primary, immune system is a reactive consequence, then that's no good for therapy. We won't do anything by blocking what's going on in the periphery. But if it's true that the B cells are contributing to anxiety, then that's much more interesting. So how can we test this? The trouble is, if you knock out B cells entirely, you get all sorts of problems with the immune system. B cells secrete things that are necessary for the formation of lymph nodes and various other things. So what we used was the CD19 knockout mouse, which has a partial deficit in B cells. So the rest of the immune system is pretty normal, but there are fewer B cells, and in particular, fewer of those regulatory B cells. And what happened? Well, we looked at the behavior, and in this mouse, by both measures of anxiety, the light, dark, and the open field testing, the B cell deficient mice are more anxious. And this is really interesting because we had no idea that the B cells could contribute to anxiety or indeed to any behavior. And why might this be? To answer this question, we looked at the meningeal immune cell compartment in these mice. And comparing it to the wild-type mice, you can see that in these mice, which just like B cells, there are now all these pro-inflammatory cells in the meningeal immune cell compartment that shouldn't be there, the neutrophils, the cytotoxic T cells. Next, what we did was we said, okay, and how does this mouse respond to stress? Repeated the experiment with the wild-type mouse replaced by the CD19 deficient mouse. How are the meninges affected by stress? As you saw before, on the left, in the wild-type mouse, stress causes an infiltration of monocytes. But in the CD19 mouse, we also get these pro-inflammatory neutrophils coming in. And looking in the spleen, basically the response was the same, apart from this. The wild-type mouse are able to increase their regulatory B cells in response to stress, but the CD19 mice couldn't do this. So what have I shown you in the animal model so far? Firstly, I've shown you we can measure changes in all these immune cell types, that stressed animals have changes in both the peripheral and the central immune cell compartments. And then using the knockout mouse, I was able to show you a causal role for B cells in driving anxiety, possibly by creating a pro-inflammatory environment in the meninges. But none of us treat mice much, so what's going on in humans? In parallel to these animal studies, this is early days of this work, we're also looking at the human biomarker study in the Wellcome Trust BioDep consortium. So I guess the analogous place to the spleen is the blood. We can't take splenic biopsies, but to get an assessment of the peripheral compartment, we can take blood samples and look at the peripheral blood mononuclear cells. And from the animal model, we predict an increase in B cells, and indeed, that's what we see. So in depressed patients, there's a higher number of B cells than in healthy patients. But of course, what we're really interested in is that balance between the regulatory B cells and the pro-inflammatory B cells. So I've optimized some cytokine assays, and we're going to be looking at whether that balance is impaired in people with depression. But how are we going to look at the meninges? Well, the nearest thing we can get is the cerebrospinal fluid, which flows within the meninges. And previously, it wasn't possible to look at the cells in this compartment because there were so few of them. There are only 1,000 cells per mil of CSF that you take. But using modern techniques, we can get at those cells. So that's what we're doing. We're taking 10 mils of CSF across these 5 centers, spinning to get a cell pellet, and then extracting the RNA from those cells and seeing how the CSF transcriptome differs between patients and controls. And in an exciting collaboration I've developed with the Sanger Institute, for a subset of these patients, we're actually going to separate out each individual cell from that CSF sample and take a separate transcriptome from those cells in a technique called single-cell RNA, which you may have heard of. So we kicked off with this study a month ago, and watch this space. It just remains for me to thank my supervisors, Ed and Mena, and much of the work I showed you I did at NIH with Miles Heckenham and Mike Lehman, and the antibody depletion studies I'm doing in Oxford with Daniel Anthony. So thank you all for listening, and I just wanted to say thank you to Wellcome and Gatsby and the Royal College, because I came into psychiatry hoping that this was going to happen, and it is, so it's wonderful.
Okay, questions? Yeah. Mic come to the middle. Oh, we haven't got any mics left. Got no mics? No mics, we don't. Okay, you're just going to have to shout in the old-fashioned way.
I can repeat it back, so.
Okay, that was on cortisol. How is that sort of
exactly.
Everything?
That's a really exciting question. So that's what I'm going to be doing with the B cells from the patients. When I'm looking at their cytokine expression, IL-10 and IL-6 for that regulatory effector phenotype, I'm also going to do a version where I add cortisol and see if that affects it.
Okay. Anyone, any others anywhere? Yep, in the middle.
Can you increase B cells, or can you transfuse B cells?
Yeah, you can. So there's 2 options. Either we can try to selectively deplete those pro-inflammatory B cells, and there are existing therapies that do that, belimumab, belatacept, selectively alter the ratio of regulatory and effector B cells. And then, as you allude to, we could also try to increase the number of regulatory B cells. So at the moment, they're developing this as a therapy for solid organ transplants. So you take a patient's B cells, you make them proliferate in a dish, and you give them back to the patient. So hopefully that's something we can repurpose here.
Could you recover the behavior in the mice, the tissue?
If we give back the B cells. So that's what we're doing now. So we're going to give back B cells that are wild-type B cells, and then give back B cells from an IL-10 knockout, and that'll allow us to see whether it is indeed a regulatory B cell-dependent phenomenon.
Okay, John.
Thanks for a really lovely talk and beautiful data. I wondered how specific you think the increase in inflammation is to depression/anxiety, or do you think there's a general feature of psychiatric illness?
Yeah, you're right. This seems to be transdiagnostic. Bipolar disorder, autism, schizophrenia. You get similar, slightly different, but really quite similar changes, this low-grade proliferative inflammation. We can't speak so much to whether the cellular changes are similar, but hopefully that's something that's going to come out of the data from Cambridge soon.
Okay. Anyone else? Yep. Nice. Meanwhile, think of a song from the Pirates of Penzance that we can sum you up with.
So kind of following on from that then, the mouse model, have you done any of the sort of depression type tests, so any tail suspension or forced swim tests in that model?
Yeah, so this model is known not to affect the tail suspension test, so we didn't do that. but you've asked me a question I didn't want to answer. So because one of the tests that this model is supposed to change is the social interaction ratio. So that after the end of 10 days, a mouse given a choice of an object or another mouse in a little cage, it would choose less to interact with that mouse. And interestingly, that's actually something that in the lab I'm working in, and at least 4 other labs, we can't reproduce if the experimenter is a female, which is really odd and frustrating. But so it's not a test that I pursued because there seems to be that variability.
Okay. Anyone else? Because you were so short that you have more time. Unlike your boss, by the way, who we now
see we work together, you know.
Who we now know is not just a worm lover, but he's also a dominant CD1 mouse. So slowly, steadily moving up the animal kingdom there. Very good. Okay. Anyone else? No. All right. Good. Thank you very much. That was brilliant. Well done.